Today I am here to present the topic Principles of Inheritance and Variation, the fifth chapter in 12th biology. Before going into the details of this chapter, we have to clear the points: why to study genetics, what is genetics, and why the two children of the same family differ each other, and why the identical twins are resembling each other. You might have seen in your family also, the children are differing from their parents in some of the fundamental characters, and similarly children are resembling with their parents also in most of their characters. Why this happens? You may be amazing. You might have think about it many times. So, here in this chapter we will learn why it happens. So, heredity, you might have heard this term, resemblance of progenies with their parents in most of the characters. For example, if the mother is having curly hair, the daughter is also having curly hair in some cases. Father is having white complexion and the son is also having the same white complexion. So, like this, in most of the characters, the progenies are resembling with their parents. So, resemblance of progenies with parents in most of the characters, that we will call it as heredity. And similarly, some of the progenies differ from their parents, and that phenomenon we will call it as variation. Now, when we study about these two aspects, heredity and variation, then it forms the branch genetics. So, genetics is a branch of biology that deals with heredity and variation. The term genetics that was coined by William Bateson in 1906, and the father of genetics, Gregor Johann Mendel. As I told you in the introduction, what is heredity? The tendency of the progenies to resemble with their parents in most of the characters. And if the progenies differ from their parents, then that phenomenon that is known as variation. Now, before going to the details, I would like to present a life sketch of Mendel. He was born in 1822 in a peasant family in Moravia in Austria. He became a monk in the Augustinian monastery at Brunn in Austria in 1843. Later, he was ordained as a priest in 1847 and he became a teacher also. He conducted hybridization experiments on pea plants at his monastery garden from 1856 to 1865. He published his findings in the year 1866 in the annual proceeding of Natural History Society of Brunn. His laws form the basis of science for genetics, so he is known as the father of genetics. He attended courses in physics, maths and science in Vienna University. He conducted hybridization experiments or breeding experiments in Garden P at his monastery garden from 1856 to 1865. But his work remained hidden for a long period of time. Why it happens? Why his work remained unnoticed for a longer period of time? His work was ahead of his time. His work was published in a journal that has a limited circulation at that time. He himself was not sure about his findings. He followed mathematical approach in working out the biological problems, but it was very strange at that time. All the biologists were busy with the Darwin's theory of evolution. So his work remained hidden for a long period of time. But later in 1900, his work was rediscovered by a group of 3 scientists: Hugo de Vries of Netherlands, Karl Correns of Germany, Erich von Schermach of Austria rediscovered the valuable Mendel's work in 1900. You can see here the pictures: Karl Correns, Hugo de Vries, Erich von Schermach, the 3 scientists who rediscovered Mendel's work. Mendel selected the experimental material, that is Garden P. Its scientific name is Pisum sativum. Why he selected a pea plant as his experimental material? Even though there are so many flowering plants with fragrance, beautiful colors, huge sized flowers, but why he selected this particular plant, Garden P, for his hybridization experiment? You all know it is a herbaceous plant, very small plant with a very short life cycle. Within 1 year it will produce its flowers and it will perishes. So it is easy for us to conduct hybridization experiments and we will get the results very quickly. The second reason is availability of a large number of varieties. Innumerable number of varieties of pea plants are available. So any scientist can use any of these varieties for his experiment. Then this plant will produce a large number of seeds. The availability of a large number of seeds is yet another reason for selecting pea plant as his experimental material. So if a plant is having only one seed, sometimes it will not germinate. So in that particular case, the result of the hybridization experiment was not obtained. So here there is no chance for this flop of the hybridization program, because large number of seeds and thereby large number of progenies will be produced. Presence of a large number of contrasting characters. Now, here I would like to mention what is a contrasting character. Different expressions of the same character, that is called as contrasting character. For example, if we are considering the character flower color, then purple as well as white, the two extremes, they are the contrasting characters. Then this particular flower that possesses, it is a bisexual flower which have self pollination. Pollen grains of the same flower will fall in the stigma of the same flower, self pollination. It is bisexual in the sense that it has both male as well as female sex organ in the same flower. Now, when you see this flower, its corolla is a peculiar one, that is papilionaceous corolla. Now, I will show you the diagram of a papilionaceous corolla. It is simply known as butterfly like corolla. There is one standard petal of large size, then there are two wing petals, and there are two keel petals. So, this particular type of corolla, that is easy for doing the hybridization experiments. So, here you can see the standard petal, large one and outer petal, and on either side there are two wing petals which are kept inside to the standard petal, and there are two keel petals, and inside this keel petal there present the sex organs, that is androecium as well as gynoecium. So, it is easy for the scientist to do hybridization experiments here. It is easy to open the petals and do the cross pollination, then we can close it also. So, that is the advantage of having this particular papilionaceous type of corolla here. Here you can see the pea flower, garden pea flower, purple as well as white. As I mentioned earlier, these two are the contrasting character for the color of the flower. The single trait, color of the flower with the two different extreme expressions, that is one purple and the other is white. And here you can see the round shaped seeds. Now, we can familiarize some of the important terms in this one. First, it is very important to know what is a gene. As we all know that gene is the unit of heredity, it is the unit of inheritance of characters from parents to offspring, it is the unit of transmission of a character from parents to offspring, which is a part of the DNA that is present in the chromosomes seen in the nucleus of a cell. And each and every character of an organism is controlled by a pair of genes. Now, the next important thing is alleles, or at another name is allelomorphs, the alternative forms of the same gene controlling the same trait or the same character which is seen in the same locus of a homologous chromosome, that is known as alleles or allelomorphs. For example, when we consider the character tall, when we consider the character height of the stem, their alleles are capital T and small t. Capital T allele controls the contrasting character tallness, and small t controls the contrasting character dwarfness. So, capital T and small t are the two alleles controlling the same character, length of the stem or even height of the plant, which are seen in the same locus of the homologous chromosome. Now, coming to the phenotype. Phenotype refers to the external character of an organism for a particular trait, or it is the external appearance, or it is the expression of a gene product. It is controlled by the genes as well as the external environment. Now, coming to the genotype, it refers to the genetic makeup or even the genetic constitution of an individual for a particular trait. By seeing a particular individual, we can say only the phenotype, not the genotype. We can never say that if a tall man is walking through the road, we can't say that capital T is walking, or we can't say that capital T small t is walking. We can say only tall man is walking. So, we can say only the phenotype, not the genotype. Now, coming to homozygous, an individual that bears two similar alleles at a given locus for a particular character. For example, capital R gene that controls the phenotype red color of the flower. Now, regarding heterozygous, an individual bears two different alleles for a particular character and seen at the given locus. For example, if an individual is heterozygous tall, then it will be represented as capital T small t. And for red flower color, if the plant is heterozygous, then we will write it as capital R small r. Now, regarding pure line or pure breeding or true breeding, an individual that produces same type of phenotype, same type of characters in all the progenies in all the generations, then it is pure breeding or true breeding or pure line. And such individuals will be always homozygous. So, later in this chapter there will come so many problems, genetics problems, there it will be given only as pure line or pure breeding. So, here the concept must be clear, a pure line plant or an individual that will be always homozygous, and it will breed the true, it will produce the same phenotype for all the generations in all the progenies. Now, how can we convert a plant or an individual into a pure breeding one? We can do continuous inbreeding in the case of animals, and in plants we can do continuous self pollination. So, by continuous self pollination or by continuous inbreeding, we can convert an individual into a pure breeding one or a pure line or a true breeding one or into a homozygous one. Now, how a character can be symbolized? Symbolization of characters. The first letter of the character, dominant character, that is taken, we will consider the first letter of the dominant character. For example, the character is tall. Tall begins with a T So, we will take the first letter T and it is written as capital letter for dominant character, and the same letter T, if we are writing in small letters, it will form the recessive character. So, this is one system of symbolization where we are using the first letter of the dominant character and it is written in capital, then it will form dominant character, and if it is written in small letter, then it will form the recessive character. There is yet another method of representation also, where we can select the recessive character, then the first letter of that recessive character is selected. It can be written as capital for dominant character, and if it is written as small letter, then it will form the recessive character. Here you can see the condition. We will take the character, contrasting character dwarf. This is the recessive character. It begins with the letter D, that first letter we are considering, and we are using capital D for representing the dominant character tall, and we are using small letter d for representing the dwarf, that is the recessive character. So, there are two system of symbolization of a particular character. Now, before going to this monohybrid cross, what are the characters studied by Mendel that we have to learn? Mendel studied 7 pairs of contrasting characters. First one, length of the stem or even height of the stem. Here the contrasting characters are tall and dwarf. Second character is color of the flower. Here the contrasting character is purple and white. Now coming to the third one, position of the flower. It is axial and terminal. Color of the pod, there comes green and yellow. Then shape of the pod, there comes inflated and constricted. Color of the seed, yellow and green. Shape of the seed. here comes round and wrinkled. So, here you can see the 7 pairs of contrasting characters studied by Mendel. Mendel considered only one character at a time and studied the inheritance of a particular character fully at a time. So, the different 7 pairs of contrasting characters that each and every child should learn and try to identify which one is dominant and which one is recessive. It is very, very important in this context for doing the genetics problems. So, the first character here is length of the stem. The different expressions you can see here, one is tall and the other is dwarf. That means, same character or the trait is expressed in 2 different alternative forms. That is, one tall and the other is dwarf, contrasting characters. Here the first one, tall is dominant and dwarf is recessive. Second character studied by Mendel is color of the flower. The different expressions are purple and white. The 2 extreme contrasting characters, expressions, phenotype. Here purple is dominant over white. In some context, in some of the genetics problems you can see red versus white. Then red is dominant over white. Now coming to the position of the flower, axial and terminal. Now what is axial? You know, the angle between leaf and stem, that is called axil, and if the flower is seen in the axil, then that flower is known as axial flower, and if it is seen at the tip of the stem, then it is terminal flower. Correlate this one as, or memorize this one, which one is dominant and which one is recessive. You can use the correlation. In your alphabet, A comes first. So, A is dominant, that means axial is dominant here, and the other character you know, that is terminal. Now, after the flower there comes the fruits, that means pod. Here we will say it as pod. Color of the pot, you know, the prominent color in plants, that is green. And here also the thing is, again you can apply, G comes first than Y. So, G is dominant, that means green is dominant and yellow is recessive. Now, in the pod, the next character is its form or shape of the pod, inflated and constricted. Inflated means external appearance, you can't see any position of the seed, where you will have the doubt whether it has seed or not. It will be just like thin and flat. And in constricted pod you can see it clearly, an external appearance of the seed can be felt outside itself, constrictions are there. And when we consider this one, capital I as well as C, I is longer one. So, you can again correlate it as the dominant character, inflated one is dominant. Now, coming to seed. Seeds are seen inside the pod. So, next character we will say the seed, that means color of the seed, just reverse of the color of the pod you can see here. So, first you memorize color of the pod, then only comes the seed. So, just reverse of this case you can apply here, that is the memorization in this aspect. So, color of the seed, the dominant character will be yellow and recessive character will be green. Now, coming to the shape of the seed or form of the seed. Form of the seed is round and the other one is wrinkled with the wrinkles. Here there is no wrinkle at all, it is smooth surface, round and prominent one. So, round and wrinkled. Here also you can see which letter comes first. R will come first in your alphabets. So, here round is dominant over wrinkled. So, in this way you have to memorize all these 7 pairs of contrasting characters. It is highly essential to learn the genetics and its problems properly. So, no need to by heart it, by correlated study you can memorize all these things. Just step by step you have to move, analyze a plant. First you can see the height of the stem, length of the stem with a tall and dwarf, color of the flower, purple and white. Then coming to the second character of the flower, that is position, axial, terminal. Then color of the pod, green and yellow, shape of the pod or form of the pod, inflated, constricted, color of the seed, just the reverse of color of the pod, that is yellow and green. Then form of the seed or even shape of the seed, round and wrinkled. Now, we will move to the next aspect, the basic of this one, genetics, monohybrid cross. Mono means single, a cross involving 2 parents, and these 2 parents are differing in 1 pair of contrasting characters. So, a cross involving 2 parents differing in 1 pair of contrasting characters, that is called as monohybrid cross. For example, we are using the character here length of the stem or height of the plant, where the contrasting characters are tall and dwarf. If we are using the character color of the flower, then the contrasting characters are purple and white. So, these 2 examples, we can make so many examples out of these 7 pairs of contrasting characters. So, this example where we are using only 1 pair of contrasting character, 2 parents are involved and they are differing in only 1 pair of contrasting character. So, it is an example of monohybrid cross. Now, Mendel's detailed hybridization experiment. First, selection of pure breeding parents. As I have already told you, pure breeding means true breeding or pure line. That plant is homozygous in genotype and it will produce only the same phenotype for all the generations. It breeds true in its phenotype. So, we can consider a true breeding plant or a pure line plant for the hybridization program. Suppose it is not available, we can convert it into a pure breeding plant by continuous self pollination. And pure breeding plants will be always homozygous, that is at another concept that we have to memorize, and they will pass the character as such to the progenies, to the offsprings. Second step, hybridization of the selected parents. Mendel selected in the original cross, the tall plant is taken as male parent and dwarf plant is taken as female parent. Before doing the hybridization experiment, we have to cover the flowers in order to avoid contamination with foreign pollen grains. Then collect the pollen grains from the tall male plant and dust it on the stigma of the dwarf female plant. That means we are doing hybridization or even cross pollination. After doing the cross pollination also, we have to bag the flower, the cross pollinated flower, in order to avoid contamination with foreign pollen grains. Now, after maturity, collect the seeds and sowing the seeds. After that, the hybrid plants are obtained. Mendel analyzed the plant in the first generation, and it was found that all the plants of the first generation were tall in phenotype. Now, here it is written as F1 generation. 1 means first and F stands for filial. The first filial generation. Now, what is the meaning of the word filial? Filial refers to offsprings, progenies. So, the first hybrid progenies are produced by the crossing of P generation. P generation means parental generation. The symbol F1, that is the first filial generation. F1, the term was coined by Batson and Sanders in the year 1902. In the third step, Mendel conducted self-pollination in the F1 hybrid plants, and in order to produce the F2 generation. F2 here means second filial generation. That means the generation in which the second hybrid progenies are produced by the selfing of F1 progenies. Mendel observed in the F2 generation 3 tall, that means 75% of the F2 progenies were tall in phenotype and 25% of the F2 progenies are dwarf in phenotype. Again he conducted self-pollination among the F2 progenies and produced the F3 generation. The third generation, third filial generation. In the F3 generation, the result obtained was amazing. 1/3 of the tall progenies produced only tall progenies, and 2/3 tall progenies of the F2 generation produced both the tall and dwarf progenies in the ratio 3:1. Again 1/4, that means out of the whole progenies of the F2 generation, 25% they are dwarf, and they produced only dwarf progenies in the F3 generation. So, listen, once again I am repeating the observation of F2 monohybrid cross. See the results of the F2 generation. You can see here 3 tall and 1 dwarf progeny. Selfing of the F2 progenies to produce the F3 generation. And here in the F3 generation, 1/3 tall plants produced tall plants only, and the 2/3 tall plants produced both the tall and dwarf in the ratio 3:1. 1/4 of the F2 progeny, that is the dwarf progeny, produced only dwarf. As I have shown to you in this Punnett square, and after that we are coming to the conclusion, the phenotypic ratio is 3:1, as we got 3 tall and 1 dwarf in the F2 generation. Genotypic ratio is 1:2:1, that is 1 homozygous tall, 2 heterozygous tall and 1 homozygous and recessive. Again 1 capital T individual, 2 individuals are having capital T and small t, 1 individual is having small t. So, the genotypic ratio is 1:2:1. Now, it is the time for you to answer the questions, the review questions. First question for you: how can we convert a bisexual flower into a female flower? Yes, your answer is here. We can convert a bisexual flower into a female flower by removing the anthers before dehiscence. You know dehiscence, that is splitting up of the anther at the time of maturity to release the pollen grains. So, removal of anthers before dehiscence from a bisexual flower, that is called as emasculation, and it is done to convert a bisexual flower into a female flower, and its role is to prevent self pollination. Now, here is your second question. We have given different set of characters here. You have to identify which among the following is dominant and recessive. Here the characters are axial flower, wrinkled seeds, red flower, constricted pod, yellow seed and green pod. You have to identify which among these is dominant character and which are the recessive characters. Yes, your answer: axial flower, red flower, yellow seed, green pod are dominant. Wrinkled seed, constricted pod are recessive. Now, it is the time for you to ponder about it or think it over. Is there any need for emasculation in a pea plant that is considered as male parent? If not, why it is so? Thank you.